Okay, everyone, thank you very much. I think we'll get started since it's 5 o'clock and there's a lot to see here. And feel free if you'd like to move forward. I promise I'm friendly. It is a little bit of a small screen, so no problem there. Thank you everyone for joining us today. My name is Courtney Anderson. I'm Director of Product Marketing at Deepcell. And it's my great pleasure to show how you can get started getting deep cellular insights through high-dimensional, high-content morphology that is AI-powered, label-free, with single-cell imaging and capture on the Deepcell platform. So it's well known that cell morphology is indicative of phenotype and function. For instance, here we can see a homeostatic microglia. This morphology indicates a very healthy environment and a surveillance state. However, reactive microglia respond to a trigger in the environment, and they release pro-inflammatory cytokines, proliferate, and change their morphology accordingly. Now, if there's neuronal degeneration, now you can see a very dramatic change in the phenotype. They alter their morphology, and then this can result in phagocytosis. Now, these differences are very clear and striking to the human eye, but for a case in this example, it's really not so much. These are two cells. One is drug resistant and one is drug sensitive. And it's very difficult by eye and looking at the morphology to really tell them apart. And that's because cell morphology is highly dynamic, it's highly complex, and analyzing by eye can only tell us so much. And so this demonstrates the strong need for a more quantitative, high-dimensional, unbiased approach to assessing cell morphology. So in order to unlock deeper biological insights from cells, we believe that cell morphology needs to be turned into a quantitative, high-dimensional ohm, the morpholome. This would be similar to how methods have been developed for the genome, transcriptome, and proteome to assess those molecular analytes at high dimensions. Now at Deepcell, our approach to morpholomics is to provide single-cell, high-dimensional, unbounded morphology analysis. And we offer in our platform high-resolution bright field images of single cells, foundation deep learning models for high-dimensional feature analysis, label-free characterization and sorting of cells, and an interactive software to store, visualize, and analyze those single cells and that high-dimensional data all in one. Now, until now, methodologies to assess and characterize cell morphology have really been limited to either imaging or sorting with labels. But Deepcell takes the best of these worlds and brings them together in one platform and builds upon that in order to provide an imaging and label-free sorting in one platform. So the Deepcell instrument, it's a benchtop instrument. It takes real bright field images that are high resolution of each individual cell. And then therefore you don't need another imaging system to do this. You can do it all in the instrument. In addition, this Deepcell instrument will also collect cells and sort cells in 6 ways based on their morphology, and it does not require any labels or staining. Furthermore, you can collect these cells. They still remain unperturbed and viable for downstream analyses such as single-cell RNA sequencing, and I'll show you some of that data in a little bit. Now, the Deepcell AI models measure both known and novel morphology parameters in real time. They also leverage very large databases, so they're continuously learning and improving insights. And then finally, the Deepcell software suite enables real-time data visualization and analysis from the cell images that are obtained on the Deepcell instrument. And the workflow is very simple and streamlined, starting with dissociation of cells into a single-cell suspension, followed by imaging of those cells in flow, and then characterization of those cells in real time with our AI models. And then all of this component is done on the instrument. In the Deepcell software suite, you can analyze the data, but you also can go in and select your cell populations of interest in real time and then collect those cells for further downstream analyses. And really, you can go from sample to publishable figure in just a matter of hours. Now, here's an example of the kind of data that you can expect from the Deepcell platform. Now, the Deepcell AI technology has 2 key components. It extracts features from the cell images of diverse human cells without prior knowledge of any kind of specific cell marker, cell type, or cell preparation. And those 2 key components are the AI or deep learning derived features, of which there are 64. These are based on deep learning models that are derived from neural networks. And then the morphometric features, of which there are 55, are the more quantitative analyses based on biological features such as size, position, and pigmentation. And now the AI models enable meaningful clustering. And what we mean by that is that they can decide and determine the cells that should be together, are they together? So for instance, here in this morphology-based UMAP, you can see the NSCLC population, those are the lung cancer cell lines, are all clustering near each other, while the hepatic cancer cell lines, the HCC ones, are all clustering together. So they cluster by their tissue, but then they also cluster by the fact that they're carcinomic, versus the cell types on the bottom, which are non-carcinoma, including PBMCs, they all cluster together as well. And so really the Deepcell's AI model has been developed on tens of millions of cell images, and as I mentioned, it's continuously learning with added data as we keep adding to it. And so this also allows for really robust and speedy training of any custom models that would be made. And for more information, I do highly recommend that you visit our AI models poster, and there'll also be a flash talk on Tuesday as well by our head of data science. Now, as I mentioned, the instrument does sort the cells. And this sorting process is gentle and label-free, which basically means that the cells are minimally perturbed and viable at the end of the process. And so here I want to show you an example of that. These are human neutrophils. They were isolated from whole blood, and then we had them undergo 3 conditions. They either were left unperturbed, they were either stained and sorted by FACS, or they were left unstained but then sorted by the Deepcell platform. And then we performed bulk gene expression profiling by RNA-seq. And what you can see here on the top portion of the graph is that the cells that were sorted by FACS have very high differentially expressed genes, either up or down regulated compared to the unperturbed cells, versus the cells that were sorted with Deepcell have minimally perturbed cells at the gene expression level. So this shows that in fact, while FACS could significantly induce gene expression misregulation, the Deepcell platform minimally perturbed the cells. And then in addition, let me just click here. We did a second experiment to assess the downstream analyses. So we wanted to see, once we've collected the cells by morphology on the Deepcell platform, could we do single-cell RNA sequencing, and what did that gene expression profile look like? So we took PBMCs, and we either just left them stored in RPMI media, or we sorted them on the Deepcell platform. And then we followed this up by 2 different single-cell assays. We either did targeted RNA-seq with an immune panel, or we did whole transcriptome single-cell RNA-seq. And what you can see here in this UMAP on the left is that there's basically overlap between the pre-sorted cells that were just stored in RPMI or the cells that were stored in, that were sorted by Deepcell. In addition, if you look at the correlation plot on the right, we see that there's near perfect correlation between either being left in the RPMI media or being sorted on Deepcell. And so this really shows that there's high correlation and no effect on the downstream analyses due to the Deepcell platform. So this allows you to then continue on and do single cell analysis and other analyses with your cells. Okay. So now with that, I've introduced the product and some of the technology, and now I'd like to show it in action and how it's been used in research areas. And the first couple of data sets I want to highlight are actually taken with our newly released Deepcell software suite demo that's publicly available now. So first, I have a melanoma data set here that I wanted to highlight. We wanted to recapitulate the heterogeneous tumor microenvironment. So we combined a mixture of human melanoma cell lines as well as primary tumor samples and then ran them on the Deepcell platform. And then using only morphology, we were able to identify a number of cell types in the tumor microenvironment, including stromal cells, immune cells, and then the melanoma cells. But then we also utilized morphology embeddings, UMAP, that was colored by Leiden clustering. And then in this case, when we dig deeper, and these are bright field images taken off of the Deepcell platform, we can see immune cells cluster together as expected, but they do seem to have some subtle and separable morphologies. And then we can also see, more interestingly, that pigmented melanoma cells distinctly cluster separately as well. And this was all done without any kind of labels, which is quite challenging with other methodologies where you do need a label. Here, this was all morphology-based. And so I want to dig a bit more into this melanoma population. So here we took the melanoma cells that are shown here in this morphology UMAP in green, and we reprojected them onto a new UMAP. And when we do this, we can get a more separation or identification of those subpopulations of melanoma cells. There's the dissociated melanoma biopsies, the melanoma cell lines, SK-MEL1 and 2, and then there's also the pigmented melanoma cell line, MNT1. And what we wanted to do is hone in a bit more and look at that latent clustering morphology embedding UMAP. And when we look at it from this perspective, we can clearly see that pigmented melanoma cell line, while it's one cell line, actually has two distinct subpopulations of its own based on morphology. And so we wanted to kind of look at that a bit more. And when we looked at the individual cell images from those two different populations, we indeed do see that there are some subtle differences that the AI models and morphometric features pick up on. So shown here, you can clearly see the green non-pigmented cells have a distinct appearance, and then the pigmented cells have more granularity to them. But even among those pigmented cells, as I mentioned, we can see differences between the pink cluster 6 and the blue cluster 9. In particular, that pink cluster 6 has what's known as blebbing on its cell membrane, and that could be indicative of these cells undergoing apoptosis versus the other cells in cluster 9 may not be undergoing that. So we could definitely see that distinction by morphology. And then the other beauty of the technology is that we can actually get a quantitation behind this. So this is showing the mean density pixel, which will help be an indication of pigmentation and granularity in the cells. We can clearly see here that cluster 2 is non-pigmented. We're not picking up with the mean pixel density, but we can clearly see clusters 6 and 9 are quantifiably different in their pigmentation. And so I do encourage you, we have several more data sets online. If you're interested in this, please check out exploredata.deepcell.com. It's publicly available, and you can just go and check out the data and also see some of the features of the software suite for yourself. Now, the Deepcell technology can be applied in a number of different ways, but two broad categories to think about are at the sample level and at the cell level, and I'll show a few examples from each of those right now. So first, I wanted to highlight a recent preprint that came out from John Yu Rao's lab at UCLA. They were really aiming to enable better precision medicine in the field of cytopathology. But they wanted to use high-dimensional cell morphology. And this is because typically in cytopathology, the methods, the types of, especially the samples they were working with, malignant effusions, actually have very low tumor abundancy, and they are limited to high background. So because of the high background and the low tumor cell number, you end up having a lot of noise in the system, and you really can't get highly sensitive detection of those rare cells. And so they wanted to see if by sorting and enriching for the carcinoma cells using Deepcell and by morphology, if they could get a more rich population of cells to look at for their patients. And so that's indeed what they did here. They have two patients, A and B. They could clearly see with this the Deepcell platform that you do indeed have clustering of the patient carcinoma cells in one area, that's kind of the orange and red. And then the non-carcinoma cells, which are the histiocytes, leukocytes, and so forth, they cluster in the pink and blue areas. So they are distinct between the carcinoma and non-carcinoma cells. What they also observed is because you see the pink and the brown or red clusters are truly distinct, that means that there's interpatient heterogeneity between these cells. So that's really interesting as well. You wouldn't expect carcinoma cells by morphology to potentially be totally different between patients, but that's indeed what they see. And then when they looked at particularly patient A, that's what's highlighted here, they did notice differences in the granularity and the texture of patient A's carcinoma cells compared to patient B's. And now, as I mentioned earlier, typically cytopathology samples, particularly the effusions, are limited by that low tumor cell abundance and high background, and that's very evident in this data example here. So what they did before Deepcell is they had their patient effusion samples, and they did a CNV analysis. And what you can see here on the top is basically flatline. They could not pick up any clear alterations in the amplification or deletion pattern. But after enriching on Deepcell for those carcinoma cells that are rare, they can clearly see these more subtle and distinct differences now because they've enriched for that population. And I think even more exciting is then they did targeted mutation sequencing analysis on 7 patients. And what they observed is that in 6 of these 7 patients, they were able to get profiles of mutational analysis, and they had higher sensitivity here. So they could see a striking improvement in that variant allele frequency detection after they sorted on Deepcell and enriched for the carcinoma cells. And then even more striking is that in 3 of these samples, they had less than 1% of that allele frequency detection. But after enriching with Deepcell, they were able to improve this from 15 to 47% in these samples. So they were really able to get more insights into these actionable molecular lesions and potentially inform treatment options for these patients. And now another key application area, because this is single-cell morphology, is drug screening, examining the effects of drugs on cell morphology, and then being able to identify cells that will be sensitive or resistant to a drug. And so shown here is an ovarian cancer cell line. It was treated with either chemotherapy drug 1 or drug 2. And we were able to run through the Deepcell platform and through high morphology analysis, show and discriminate drug-sensitive and drug-resistant cells. And that's reflected in these differential density distribution plots that you see on the bottom that are highlighting these drug-resistant cell populations compared to the parent cell line. And now the final data example I wanted to highlight here was to show the single-cell resolution. Because we can take that single-cell high bright field imaging, we can also phenotype at this level. And this can help us really assess the health status of cells. So here we're showing Jurkat T lymphocytes. They were treated with either heat shock or staurosporin, and then they were run through the Deepcell platform. And from this UMAP taken by Morphology and overlaid with images from Deepcell, you can clearly see that there are distinctions in morphology depending on the type of treatment that the cells underwent. Now, we wanted to see how these cell health statuses by morphology compared to traditional methods. So we looked at this by flow, and you can clearly see here on the top row, this is flow cytometry of those Jurkat T lymphocytes, treated same conditions, and then they were also stained with markers to indicate viable, dead, apoptotic, or necrotic cells. And now we can see here with treatments 3 and 4, that treatments 3 and 4 basically identify a population that's necrotic. But if we took the Deepcell platform method, we actually see that these 2 treatments reveal 2 distinct populations. They're not clustering in the same manner. So by morphology, they actually are different. They could be at different stages of necrosis, at different points along the progression to cell death. So we're able to get more rich and subtle information by using the high-dimensional morphology. Now, I've just touched upon a few examples of how Deepcell has been applied, but we've seen it used in a number of key research areas, including cancer, developmental biology, cell therapy, and more. And so I hope I've convinced you today that deep cellular insights start here with the Deepcell platform, which is a fully integrated solution for single-cell, high-dimensional, unbounded morphology analysis. And while we're at the end of before I get to that, I wanted to point out too that we'll be launching the platform later this year, but we do have 2 ways to access the Deepcell technology starting today. We're very excited to announce the launch of our Deepcell Applications Lab, where you can send your samples directly to Deepcell and we'll run the entire process for you. With this, you would receive imaging data for up to 4 samples as proof of concept to support grant applications. And if you're interested in this offering, please visit us at our booth or email the address on the screen here. And then I'm also excited to announce the launch of our scientific challenge, where you can enter an application to win a free proof of concept project that's performed all by the Deepcell Applications Lab. And we're accepting submissions starting today, so just check out this QR code for all the details, and you can submit to win a free project for up to 12 samples. And the deadline is through April 26th, so you have some time to think about your project. But with that, while we're at the end of my presentation, there is so much more Deepcell at SLAS this year. First, we're at booth 446, which is literally just around the corner, so please come visit us at the end. We would love to meet with you individually. And then we also have a number of poster presentations on various aspects of the technology. One poster that's on the entire platform, one that shows those downstream analyses with single-cell RNA sequencing. And then I also like to highlight our deep learning models poster, in which we'll have a presentation at 5:20 on Tuesday by our head of data science, Kevin Jacobs. And so with that, I'd just like to thank many of our collaborators that are around the world, as well as all of you for joining us today. And we look forward to hopefully having more conversations with you. So thank you very much. And I'm happy to take questions if we have time. I'm not sure. Okay. Feel free to find myself. And again, please come to booth 446 if you have any questions. Thank you.